السلام عليكم ورحمة الله وبركاته. سعادة الأستاذ الدكتور عبد الوهاب زايد، أمين عام جائزة خليفة الدولية نخيل التمر والابتكار الزراعي، السادة الضيوف الكرام داخل وخارج دولة الإمارات العربية المتحدة، السلام عليكم ورحمة الله. نتشرف أن يتجدد اللقاء بكم في الندوة العلمية الافتراضية الأولى التي تنظمها جائزة خليفة الدولية نخيل التمر والابتكار الزراعي للموسم الثقافي الثالث 2022. قبل أن نبدأ الندوة نلفت عنايتكم إلى أننا سوف نضع جميع الحضور على وضعية الصامت حرصاً منا على جودة المتابعة والفائدة، ومن لديه سؤال يمكن أن يطرحه مباشرة في نهاية المحاضرة من خلال خاصية رفع الأيدي المتاحة عبر المنصة الإلكترونية. كما نرجو من جميع الحضور أن يبادروا إلى إدراج بياناتهم عبر الشات، الاسم، رقم الهاتف والبريد الإلكتروني، وذلك من أجل أن نرسل لكم غدا كما عودناكم العرض التقديمي للمحاضرة بصيغة PDF. والآن ندعو سعادة أمين عام الجائزة الدكتور عبد الوهاب زايد ليتفضل بإلقاء كلمته.
بسم الله الرحمن الرحيم. أصحاب السعادة، ضيوفنا الكرام، الإخوة الباحثين، الخبراء الفنيين المختصين بزراعة النخيل وإنتاج تمور والابتكار الزراعي، الأخ العزيز الدكتور مصطفى عيدشيت المحترم، السلام عليكم ورحمة الله تعالى وبركاته. باسم الأمانة العامة لجائزة خليفة الدولية لنخيل التمر والابتكار الزراعي نرحب بكم أجمل ترحيب عبر هذه المنصة الافتراضية، ويسعدني أن أنقل لكم تحيات معالي الشيخ نهيان مبارك آل نهيان، وزير التسامح والتعايش، رئيس مجلس أمناء الجائزة. في هذه المحاضرة كما أشار الأخ عهد الأولى ضمن سلسلة المحاضرات العلمية الافتراضية للموسم الثقافي الثالث التي تحظى ثالث سنة 2022، والتي تحظى باهتمام ودعم سمو الشيخ، حيث تأتي هذه المحاضرة بعد انقطاع دام عدة أشهر بسبب تحضيرات حفل تكريم الفائزين بالجائزة بدورتها الرابعة عشرة، وكذلك بسبب تنظيم المؤتمر الدولي السابع لنخيل التمر الذي حضره أكثر من 475 باحث وخبير دولي في مجال زراعة النخيل وإنتاج تمور، وما رافق هذا الحدث من اجتماعات ومبادرات كثيرة ومختلفة، معظمكم شاهدها عن قرب أو شهدها عن قرب في قصر الإمارات بأبو ظبي. ونزولاً عند رغبة السادة الزملاء الباحثين والأكاديميين أعضاء جروبات الواتساب، وتفعيلاً لاقتراح الزملاء، وهنا نشكر مبادرة الأخ العزيز الدكتور عبد الباسط إبراهيم في هذا المجال، تتشرف الأمانة العامة للجائزة أن تطلق سلسلة من المحاضرات العلمية الافتراضية المتخصصة في مجال الزراعة النسيجية وإنتاج فسائل النخيل نسيجية وما رافقت هذه الصناعة من تحديات، حيث تعمل على استضافة عدد من الخبراء والمختصين في المجال على المستوى الإقليمي والدولي. ونتشرف أن تكون البداية مع أحد الخبراء في مجال الزراعة النسيجية. لقد أخذت الجائزة على عاتقها أن تنظم 6 محاضرات في هذا المجال، ومحاضرة اليوم هي الأولى في هذه السلسلة. وحرصا منا على تعميق الفائدة، نرجو من الإخوة المحاضرين والإخوة الحضور الابتعاد عن ذكر الأسماء والعلامات التجارية للشركات أو المختبرات المنتجة لفسائل النخيل النسيجية، لا بالسلب ولا بالإيجاب، لأننا مؤسسة علمية حكومية لا نهدف إلى الربح، كما لا نهدف إلى الإساءة إلى أحد، فنحن على مسافة متساوية من الجميع، متمنين أن تقضون معنا وقتا مفيدا، ونعدكم بالمزيد إن شاء الله. والآن ندعو الأخ العزيز دكتور مصطفى أيتشيد لتقديم المحاضرة. معك أخي العزيز 45 دقيقة، فلتتفضل مشكورا.
بسم الله الرحمن الرحيم، والسلام عليكم ورحمة الله وبركاته. شكرا أستاذ عبد الوهاب. فبداية أولا أود أشكر الأستاذ الدكتور عبد الوهاب زايد، أمين عام جائزة خليفة الدولية لنخيل التمر والابتكار الزراعي، عن دعوتي كأحد المتحدثين ضمن المحاضرات العلمية التي تنظمها الجائزة. كما أشكر المهندس عماد سعد على مساعدته الكبيرة في تحضير هذا العرض. يشرفني ويسعدني أن أكون معكم جميعا هذا المساء. So the conference is going to be in English, as I said before to the organizers. But the discussion will be, of course, in Arabic, in both Arabic and English. Before starting, I would like to introduce myself. I am Dr. Mustafa Aytshid. I hold a PhD degree from the University of London on plant biotechnology. I'm currently working for a private company, one of the largest in agriculture and agro-industry in Morocco, the Domaine Agricole. I'm the technical director for nurseries and TC laboratories. I'm also honored to be the president of the Moroccan Association of Date Palm Tissue Culture Laboratories in Morocco, of course, and a vice president of the Moroccan Interprofessional Federation of Date Sector. I'm also a consultant for commercial laboratories at the international level. So my presentation is entitled Tissue Culture and Traceability System in Date Palm. This clearly means that we have two aspects that we would like to talk during this talk, which is tissue culture, of course, and traceability. So when I was preparing this presentation, I was wondering whether I should give the same importance to both subjects or areas, tissue culture and traceability. Then I realized that very little information is available on the bibliography describing really how the traceability system would work in a commercial lab producing TC plant. So maybe you will see that the presentation is more or less maybe 40-60 or 30-70. So I'm going to start the presentation now. Put it in presentation mode. Does anyone see the presentation? Okay, so like I said, I've got two aspects I would like to discuss with you or to present you is the date palm tissue culture, the technology as itself. We're starting with the advantages of TC date palms. We'll talk a bit about organogenesis versus somatic embryogenesis. Then we'll go through the phases of production and some possible improvements of the technology, whether it's through somatic embryogenesis or organogenesis. Then we'll go to the traceability system. Then we'll talk about the steps involved, the software management that can help the management of the whole production, and quality control using DNA analysis. And then, of course, finish with a conclusion. Now, what are the advantages of the tissue culture? Because the first question we might ask ourselves is to say, why do we need tissue culture in the first place? I mean, since we can grow date palm using offshoots, we all know you are all experts and we cannot use seeds, but of course we can use offshoot to propagate in a true to type manner date palm. But tissue culture plant has got a lot of advantages that do not have offshoots. I will not go through all those details because everybody knows them and we have a very high skilled expert with us. So I don't want to go through every sentence here, but the important thing is that the tissue culture plants are healthy and true to type. They are free of pests and diseases. They offer a very vigorous and homogeneous plant. So it's ideal for modern plantations. So to us, at least in Morocco, it's a must for modern plantation because then You can also propagate selected varieties or cultivars like majhul in a very large scale. You can propagate special cultivars or maize that have some special specifications. And do not forget also that the only way, the best way, if it's not the only way, really to exchange plant material between countries, because that's what gives you the best guarantee that you don't spread diseases and pests from one country to another. So let's go now through let me just reduce this. Okay, I see better. This slide is trying just to compare between the two technologies, the somatic embryogenesis and organogenesis. So as you can see, we are starting from the same kind of plant material, which is the apical dome of an offshoot, and we end up with the same result, which is a number of plantlet obtained from that offshoot. But the root is different. So if you see on top, you've got the organogenesis, where you initiate actually buds, and then you multiply those buds, then you elongate them, root them to have plants which are ready to go to greenhouse. In the somatic embryogenesis system, you don't have buds, but callus. You go through a callus, and then you've got development of these embryos, and then you've got maturation of the embryo, rooting, and then again, you've got plantlet ready to go into acclimatization. So I'm showing you some pictures at the bottom where you see clearly big difference between somatic embryogenesis and organogenesis, where you have in these three left pictures, you've got very clearly some remaining of callus and some embryos starting to germinate, and you can see the formation of embryos very clearly. Actually, the embryo is obvious because you've got the roots already starting to form, where is in the same time as the shoot. Where in organogenesis, you've got buds, you have no trace of callus. And if you do, then you are not in the right path for somatic embryogenesis. And this can happen actually if you are not careful. You might have some callus within your multiplication or initiation of the buds in organogenesis. So you see in these two pictures at the bottom that you have clearly a cluster of buds, and then you can just propagate those buds, separate them, elongate them, and have roots and have rooted plants. So I hope this picture will show you in a very brief way the big difference between the two technologies. Now let's go back to what we are using here, which is organogenesis. Very briefly going through different steps. So of course you've got a selection of an offshoot from a certified mother block. And it has to be an adult tree that already gave fruit, so you are sure that you are propagating the right plant material. Once you initiated the buds, you go into a multiplication of these clusters. So until you get a number of buds that you can start elongate, then you go through an elongation stage where you isolate every single shoot, so that it will elongate before rooting it. So of course, every step has got different conditions in terms of incubation conditions and different media also. So the end product of these steps in the lab, of course, is the rooted plant, as you can see on the left. That's a plant which is ready to go into the next phase, which is the acclimatization phase. Now, I'm not going to go into much details in terms of technology, like I said, or incubations and all that, but just few rules, very principle, very general, let's say, principles. Well, in terms of temperature, we'll find in most publications is 25 to 28 degrees. Actually, it's very much depend on stage of multiplication and the genotype. We know that majhul, for instance, is very sensitive to heat. So, but the rule is, you see on the left, is that as you go from initiation to acclimatization, usually the temperature and light intensities goes higher and higher. So you start with low temperatures and you end up with high temperatures. Again, you start with no light at all for initiation in both techniques, and you end up with high intensities of light. And it's the opposite for the media. You usually start with rich media with quite relatively high concentration of growth regulators, depending on the technology. and you end up with very light media because you're preparing your plants to go outside for acclimatization. No, of course, you can use incubators or conditions where you can control better your conditions of incubation, especially in the first stages where it can be very tricky, whether you are working on organogenesis or somatic embryogenesis. In both cases, the tricky part of the whole process is really initiating some buds in organogenesis or getting a response in terms of callus, I mean, an embryogenic callus, a callus that will eventually evolve and give you some nodules and embryos. So don't forget that high temperature will affect the media, of course, and the response of the tissue. So you never want to go higher than you need. You need alarm and notification for any excess temperature in relation with light. And there is a necessity for recording all the cycles, what's happening, because that's the only way you can know why you didn't have a reaction or why you had a problem initiating your cultures. That's in terms of temperature and the temperature, basically. Now let's talk very briefly about light. Again, you'll find so many different things in bibliography in terms of intensity and quality. So it's very important here to define things. Where we talk about intensity is the quantity of light that that the plant will receive and that the plant also need for its growth. Light quality is the type of light you will give. Are you talking about a light with the blue light or red light or both, or a full spectrum? I mean, what is the kind of quality of light you are dealing with? So this is light intensity and light quality. And of course, there is what we call photo period. So usually in bibliography, you find that most of the publications talk about 16 hours of photo period. Honestly, it doesn't mean anything because it all depends of intensities you have and of type of light you have. So that's why you have that range, 1,000 lux to 7,000 lux, depending on stage, and most of the time 16 hours for the period. So I think it's better to talk about DLI, what we call daily light integral, which is quantity of mole of light per meter square per day. So that's the amount of photosynthetically active radiation that will receive the plant during a day for a given let's say phenomenon, whatever you want, shoot or roots, whatever you want, depending on whatever you want, you will set your daily light, but also depending on intensities. So the problem is that in light, a lot of people continue to use lux because it's a convenient measurement to determine what we see as the brightness of a beam. When you look at the light, if it's very bright, you see, okay, the lux must be very high. And if it's dim, lux is probably very low because that's what you see. But actually, that's not what what affects the plant. And the plant goes for the whole spectrum from almost UV light to far red light. So it's better to talk, when we talk about photosynthesis, to talk about PPFD, photosynthetic photon flux density. Now, how the PPFD and it's related to DLI is very important, and I'll just show you an example here, where you have a publication, and they give you the reference of that publication, and when you see that long PPF with low PPF and long photo period, it's usually better than high PPFD and short photo period. In short, don't put too much light and say, okay, I will reduce my exposition to light. Instead of 16 hours, I'll go for 12, and it's going to be shorter. No, it's rather better to have less intensities, less lux, if you will, or better, less low PPFD, but increase the time of exposure to the plant, because a lot of studies showed that's the better way to do, because then the plant will have time to absorb enough photons in order to grow. And you see in that example how large the plant is in the top, and where the plant is much smaller in the bottom, where we've got high PPFDs and short photoperiod. So I think this matter of light is a big subject. Maybe one day we'll have a presentation just on this subject because it's huge. No, just going through that process, whatever it's somatic embryogenesis or organogenesis, you can always improve the technique. And I'm just giving one of the examples using, you know, what we call the TIS, the temporary immersion system. Let me get rid of this. So the temporary immersion system is an automated cultivation system based on alternating cycles where you have the tissues in the media for a given time period, and then the media goes down and the tissue stays with no media, so that you have time for gas exchange. That's why it's called temporary immersion system. And we tried in our lab this system. It's working very nicely because it's got so many advantages. It improves the multiplication rate. It eliminates any dead or brown tissue because liquid media does not allow any brown tissue to evolve more than that, or even bad colors will just die. Allow handling of bigger quantities of tissue and better structure of the plant at the elongation stage, as you can see in the picture on the right. However, there are a lot of drawbacks. Actually, I just cited 3 main drawbacks here, which is the high risk of infection. So if you manipulate a lot of plant material, it's very good because you work quicker, and how they call that, the speed of propagating plants is higher. But in the same time, you are taking the risk of affecting much more plants in one subculture. It's a high maintenance because you need to change filters, you've got tubes, you need to make sure that there is no leakage, etc., etc. So in case of loss for any reason, the impact is bigger. So it's maybe very interesting at the research level. I find it very difficult to apply in a commercial lab. No, of course, when you go through a system like this, well, a propagation system, or in a commercial lab, whether it's through somatic embryogenesis or organogenesis, you need a good facility. I need very well-trained human resources. So when we talk about facility, it means that the building is well set up from the start, and there are a lot of mistakes that people make because they take any building and start to try to establish a lab in a pre-existing building, which is usually very, very difficult to do, and it will not set the purpose. The design should be well done, and the growth room should be very well adapted. The equipment should be adapted, maintenance, calibration, everything should be followed very carefully. The human resource is very important, of course. You need to have a clear organization chart, specialization of teams, good scientific background for the manager, especially, so that he can follow the process of the production. So now that was the lab. Now what's happening once we come out of the lab? Then we have the acclimatization stage, where you can see the phases up to the field. So the rooted plant that comes out of the lab are acclimatized under greenhouse, where of course the relative humidity and temperature are controlled. Then once the plants are hard enough, they can move from small torpedo to the bigger pot, like 3 liters or 5 liters, depending on what the need of the customer. And then the developed plant can be transplanted into the field. TC palm, like we said before, are very vigorous, and usually the success rate is 100%. And if it's not 100%, there is something wrong, either with the plant in the beginning, or more to do with the way the plant is handled from the greenhouse to the field, then from the field to the plantation, to the plantation itself. So, like we said before, the plantations made of TC plant are usually very vigorous, healthy, and very homogeneous. So I'm just showing you a picture on the bottom there, where you go from the torpedo up to the plant that can deliver to the farmer. And when it's a certified plant material, and you apply or you obey the rules, then you are certified by the Ministry of Agriculture, and you are allowed to sell those plants as certified plant material. So we went through the process quite quickly. Now let's talk about this traceability system. So what it is? What is traceability system, and why you need it? Well, a traceability system is simply a system that will allow you to have all information that are recorded, and you can have access to that information anytime. The ISO definition talks also about the history. It means that at any time you can go back and have the history of any batch, any plant at any time in your lab. Why do you need to do that? Well, the ISO people will tell you this definition: increase supply chain visibility, improves quality control system, and reduce risk. Okay, but in our case in Morocco, it's a legal requirement to start with. I mean, if you don't have a system of traceability, you won't have a certified plant material. But also, of course, it reduces risk of mixing varieties, over subculturing plant, because if you don't have record, you don't know how many subcultures you've been through, a given batch was been going through. And you can also react quickly, because when you see a problem, it's only to do with one batch. You don't need to start from scratch for the entire lab. You just go and deal with the batch with the problem. So the traceability system has to go through the whole process from mother block to the field. As you can see, you've got all the process here, and they put data analysis at the bottom where it's required, at least when you take an offshoot, and at least again before delivering it to the farmer. But of course, you can do that at different stage, at any stage. That the beauty of DNA is that you can take any tissue and analyze it and see if it's true to type. So we're going through those 5 stages. Mother tree first. So in our case, we've got a mother block, as you can see, with plant here, with the offshoots, and every single tree is labeled. You've got a number for that mother tree. The ministry will give us a number, which is always with that tree and with all the offshoot that comes from the tree. There are a number of things that you have to be careful with in terms of the mother tree, and also when you are collecting offshoot. So of course, you have to make sure that it's a real offshoot, and you have to work with a single variety at a time. It has to be labeled. Each year we look at the fruit from the tree so that we are sure that it's actually the right fruit, and there is no problem with the formation of that fruit, so that the plant is real true to type. And every single plant is DNA tested before we start taking any offshoot. Then inside the lab, what we do is that we will follow every step in terms of culture, media, the operator, and the incubation. So we know which culture it is, what is the batch, what media do we have, who did it, what is the operator, and where it was stored. And so in terms of light control, PPFD, spectrum, etc., temperature control, etc. So every single tube and every single jar is labeled, and we know where it comes from and what was the history. In greenhouse, the same thing. Once the plants are coming out from the lab, we have 2 things that we have to control. The plant themselves, so we have to define the specification as the end product, which kind of plant needs to go to the greenhouse, and it's also labeled, and the conditions are also recorded. Temperature, humidity, performance criteria like loss of the during acclimatization, irrigation, fertilization, protection, etc., etc. So the risk here to control is all the identification coming from the lab to the greenhouse. You should not have, a gap between the lab and the greenhouse. Otherwise, you cannot go back to history and see what's happening in your plants. Now, if you go to the last stage, just before the delivering to the plant to the farmer, as you see, the plants still have the label there, so we can even put them again in the field and record the situation in the field, so we can follow later on the situation of every single plant. So we do that with some of the farmers, modern farmers, who do like to know the history of their plant. No, you can do that using a spreadsheet and Excel tables, but of course, it's much easier if you use a software like what I'm presenting in this case. The risk, of course, is reduced highly because the human error will be reduced, and the traceability is going to be much easier because everything is just barcoded, and the reader will just read whatever is there. And if there is a mistake, you cannot go forward. It will stop you. So what do you do actually with that software? So we just go exactly like we said. You got those 5 steps, and you do exactly the same what using the software. So in software here, you will like the introduction, the first offshoot coming from the mother tree. So it's coming with a label like you see in the picture. You've got even color coded, a color for every variety, just in case the label goes off or something. And then as soon as it comes, you put the mother tree number, where it's come, where is the mother block, and even the date, the person who collected that offshoot, the code that was given to the offshoot, etc. So you've got all the information that is recorded here. Then you put that through a line of production. I'm going to go very quickly. This is just tell you a given day, if you have that majhul, for instance, that is being subcultured, you can see in real time how much, what is the situation of that work. So you can see in that instance that that batch was subcultured. We got almost 56% of that batch was already subcultured, and the other batch you've got like 43% of the batch is subcultured. So you can follow in real time what's happening in the lab. Of course, you can also manage the stock you have, so you can see at any time what is your situation in the lab. You can also click here and see in the same majhul, for instance, what cultures are in a jar, what do you have in tube. So you have all the details at any time of your situation inside the lab. Then when you are going to deliver the plant, the system is still working. So you will scan every plant that will go for the farmer. And of course, that creates you a delivery form. So that form will be delivered only if all the criteria are met, and any plant with the wrong label or something wrong with it, it will not accept it. So you will reduce any risk of mixing varieties or putting the wrong plant in the wrong batch. So you have a delivery form that is printed automatically, and then that goes to the field. No, I was going a bit fast because I want to make sure that I keep track with time, but of course during the discussion we can come back to any of this aspect. So let's go back now to the DNA analysis. Like I said, if you remember here, you've got those 5 stages, and we say you need to make a DNA analysis at least from the offshoot that you take from the mother tree, and again at the shed house before delivering it to the farmer, so that you are sure that all your plants are true type, and also you can give assurance to the farmer that what he's taking is the right variety is taking. So very briefly, what we do in that technology, these are the steps we go through. So the best way to confirm that the TC palm are true to type is to use some DNA technology. In this instance is the SSR. SSR genotyping involves the use of sample sequence repeat that we call SSRs or microsatellite as DNA markers. That type of repeated DNA sequence is found in almost all the genomic plant, the genomic DNA in plant, and that motif of repeated sequence can amplify a profile that will allow you to recognize one variety from another. So I'm summarizing very quickly here the steps. So you receive your samples of DNA, let's say leaves coming from the offshoot that you're going to introduce into your lab. So you prepare your DNA, you grind using this machine and prepare your DNA, and then you quantify that DNA, then do for a PCR reaction, and then take that PCR product, put it through an electrophoresis machine, and then the data coming from there, inject it in the data analysis with a gene mapper in this case, so that you can analyze it and see what it gives. So these are the steps. DNA preparation is extracted from lyophilized plant tissue samples using different buffers. Then the quantity of the quality and quantity of the extracted DNA is evaluated by measuring the optical density with a spectrophotometer. PCR reaction is prepared using the appropriate markers to amplify a specific genomic DNA using a thermocycler for a number of defined cycles. Then that PCR product is put through a capillary electrophoresis that will separate the amplified fragment based on their size and the electrical charge. Then you take that data coming from the electrophoresis and analyze it using GeneMapper software and compare it to an existing database. Now, that's where the things is tricky, is actually the database. It happens to me sometimes to discuss with the people doing some analysis, and I always wonder what is the comparison you are making to compare your samples with. So unless you've got a database that was made from different varieties using a given number of markers so that you know what it looks like, you cannot compare the plant you want to try or you want to test. So let's say in this instance, you've got 3 varieties, Majhoul, Bufbus, and Barhi, and you've got 2 markers, and you can see that if you use only one marker, this is what it will give you. You see, sometimes amplification will give you the same type of amplifications, but you will have a difference in this case for Bufbus. So what I'm trying to show you here is that if you really want to have a very robust analysis, you need to increase the number of markers, obviously, because the more you have markers, the more the difference will appear, and then you can make sure that these varieties are very different, or you can make sure that these samples are really true to type to this variety. So often when we ask a lab for analysis, we have to be as a customer. Let's say when I go to the lab and they would like to ask them to do analysis, I need to be very precise in terms of what is my objective. If I've got a batch which is, let's say, Majhoul, but I just want to make sure, so I will just it's not a pure identification, it's just a verification, let's say. So I will settle for, let's say, 6 or 7 markers. It's okay. But if you got a samples and I'm not sure at all what it is, and I hope it's Majhoul, but I'm not sure, then I will go for a higher number of markers. I will go for, let's say, at least 15 up to 19 markers to make sure that I have actually the right variety. Another thing is, if you don't use enough markers, you might miss some somaclonal variations, meaning that 2 or 3 markers might not show that variation, but when you go higher, using like 10 or 15 markers, then you can see there is an allele that was amplified in that batch, and that should not have been amplified that was not amplified in the original database. So you have somaclonal variation. Now, how can we conclude? Date palm is a difficult species for sure to propagate in vitro. In order to have a steady production of high quality plant, commercial lab needs to have a very well-trained and qualified team. There is no question about that. The lab needs to be very well organized with clear procedures at every stage. Date palm tissue culture business can be very profitable in the production process, and if the production process is under control. The production cost of tissue culture is very high, so any mistake will cost you a lot. So that's why traceability and organization is important. TC date palm is the best plant material, like we said before, for establishing new plantation, thanks to all advantages that it provides, like we said before. So is the traceability system important? Yes, it's a game changer. Either you have it or you don't, but if you don't have it, there is no way that you can really convince, let's say, a farmer that your plant is right and it's true to type and is of a high quality. But if you do have a traceability system, then not only you can prove that your plant is of high quality, but also you can reduce mistake. And if there is a mistake, you can react and solve the problem. So the traceability system is a must, and very little information is available today. But people are starting working on it. Actually, I received a message from Dr. Salah saying he's working on a traceability system. He's with us in the association, which is very good. So the traceability system is something that we cannot work without in any commercial lab. Thank you very much. I hope I've not been too fast, because I was really worried not to be in time, but actually maybe I've got still something like 10 minutes to go. But I prefer to finish my talk and have time for discussion with you all. Thank you very much.
شكرا جزيلا دكتور مصطفى على احترامك للوقت وسرعتك، والحمد لله قبل الوقت. سعادة الدكتور عبد الوهاب زايد، قبل ما نطرح الأسئلة أي مداخلة من سعادتك وبعدين نبتدي نتلقي الأسئلة من المشاركين إن شاء الله.
إن شاء الله. Thank you, Dr. Mustafa. I would like to make sure that personally I thank you for such a nice presentation, full of information, for both tissue culture specialists, but also people that are away from such field. I would like just to mention a few points to make sure that the picture is clear for everybody. In Morocco, there are actually 7 commercial laboratories that are working hard, all of them, to satisfy the needs of Morocco in regard to tissue culture material. Also, there are 2 commercial labs doing the DNA analysis, being at the service of such laboratories. Third point is, without such traceability system, the producer of tissue culture material cannot sell the produced material unless there is an official certificate from a specialized agency in Morocco. And in all steps that Dr. Mustafa has mentioned nicely, there are committees who make sure that the work is done properly, and they are, for example, taking offshoot from a tree. It's not done in the classical way you go and just take an offshoot. INRA representative should be there. ONSA, l'Office National de Sécurité Santé Agricole, from the Ministry of Agriculture should be there. A representative from the lab should be there, and the owner of the farm. There is minutes and everybody sign, and such material will not leave the plantation until it has the full papers. Once it arrives in the laboratory, the lab will not dissect such offshoot unless there is a representative from the ministry who come to the lab and make sure that the data received from the southern part of Morocco is, and the type of shoot and the number and all information is respected. So this is very important. So proudly we can say that Morocco is the only country in the world doing this so far. So that's why personally, when I saw such discussion about traceability, I immediately picked up the phone and selected one of our colleagues, Dr. Mustafa Ishit, to present it to you. I am very happy to announce that, as you can see, the participant has reached 55 interested scientists, people from 14 countries. Just for reference purpose, I'm going to cite these countries. You have Morocco, Syria, Iraq, Jordan, Sudan, Spain, Egypt, Algeria, Libya, Tunisia, Iran, Yemen, Sri Lanka, and Indonesia. I'm sorry if somebody from another country I missed, but this list has been communicated to me by one of the Khalifa Ward staff. Now the floor is back to Mr. Aad for questions, please.
Thank you, Dr. Zayed. The floor is open for questions. Please, who would like to have a question should raise his hand so we can unmute him. Okay, we start with Dr. Ibrahim Jabbouri. Dear colleagues, please unmute Dr. Ibrahim.
شكرا دكتور عبد الوهاب زايد، شكرا دكتور مصطفى. الحقيقة كان لي الشرف أنه أتعرف على دكتور مصطفى، وهذه من فوائد مؤتمرات جائزة خليفة الحقيقة الدولية نخيل التمر والابتكار الزراعي. الحقيقة أنا مو مختص بزراعة الأنسجة، بس تعلمت كثير من هذه المحاضرة، وأنا يعني شغوف جدا أنه أتعلم بعد بمحاضرة ثانية إذا دكتور عبد الوهاب زايد يعني كلف دكتور مصطفى مرة ثانية أنه يعطينا محاضرة ثانية من هذه السيريز، كما هو قال أنه ممكن أن يكمل هذه المحاضرة بمحاضرة أخرى. الحقيقة المحاضرة هذه اجت نتيجة مناقشات، ويعني مهم جدا أنه دكتور عبد الوهاب أشار عدم ذكر الشركات، وإحنا ما لنا علاقة بالشركات. محاضرة علمية، وكانت يعني الستيتمنت اللي بداها بالبداية كانت يعني ممتازة، لأنه الحقيقة المناقشات اللي كانت على الجروب هي اللي دفعت إلى هذه المحاضرة، واللي خلت الجائزة الحقيقة تاخذ الانشيتيف انه تعمل هذه المحاضرة. انا ما عندي اسئلة دكتور مصطفى، بس حبيت اشكرك جدا وتعلمت من عندك كثير، وان شاء الله لما تكون يعني البرزنتيشن عندنا بين ايدينا راح نكون احنا فرحانين جدا. شكرا.
بارك الله فيك استاذ.
شكرا.
Dear Samira, please can you unmute Dr. Abdul Basit Ibrahim and open the access for everybody to unmute themselves. Thank you.
شكرا جزيلا. بداية شكري وتقديري للأمانة العامة لجائزة خليفة الدولية لنخيل التمر والابتكار الزراعي على هذه المبادرة، وشكري وتقديري لأخي الدكتور مصطفى على هذه المحاضرة الشاملة والقيمة. أنا الحقيقة السؤال راح يكون يعني ربما الدكتور مصطفى ما تطرق له، وقد ربما تكون في محاضرات قادمة إن شاء الله يتم التطرق له. الآن إحنا عندنا طريقتين معتمدة اللي هي Somatic Embryogenesis والأورجانوجينيسيس. طبعا أنا يعني دائما هذه أقولها في كل محفل، إنه الأخوة في المغرب هم المتميزون في زراعة الأنسجة، وخاصة الآن يتبعون طريقة الأورجانوجينيسيس. لكن الآن السؤال هو: هل لوحظت هناك جينيتيك فاريشن في نخيل الأنسجة الناتج من الأورجانوجينيسيس كما لوحظت أو سجلت على الطريقة اللي هي تتبع في الإنتاج الكوميرشال التجاري اللي هي بالإمبريوجينيسيس؟ وشكرا لك دكتور مصطفى على محاضرتك القيمة، ونتمنى إن شاء الله نستمع إلى محاضرات أخرى. شكرا جزيلا.
Mr. Abdullah Dr. Abdullah bin Abdullah, please can you ask your question?
السلام عليكم، أهلا وسهلا. دكتور مصطفى آيتشيت، شكرا على المحاضرة.
أهلا وسهلا.
أهلا بك. الحقيقة أنا عندي بعض الأسئلة. أولا، السؤال الأول هو أنه إلى أي مدى نعتبر الموليكولار بيولوجي تيست اللي نعملوها DNA يعني تمكننا من أن فعلا معناها نقولوا عندنا تروتوتايب. هذه الحاجة الأولى. السؤال الأول. السؤال الثاني، الحقيقة أنه من مدة طويلة ما تم يعني بعد 2006 ما عملنا ولا evaluation للحالة متاع النخيل النسيجي المنتج والمغروس بالملايين الآن. فأنا أريد رأيك، لأنه احنا الحقيقة أنا مشيت لكثير من الدول ونعرف أنه فيه، صدقني، فيه نسبة كبيرة من التغيرات اللي شفت على الأرض. يعني إن شاء الله تتاح لنا فرصة أيضا في واحدة من المحاضرات أنه نقدم، وأنا مستعد لهذا، يعني التغيرات اللي ممكن تقع واللي موجودة الآن. السؤال الأخير والثالث هو: ما تقديرك لمدى يعني نسبة تأثير البيرسونال في اللابوراتوري على هالتغيرات هذه وعلى المشاكل؟ لأنه أنا نتذكر في 2006 خرجنا بكونكلوجن هو أنه التقنيات الاثنين Organogenesis والامبريوجينيسيس يعني أعطوا معناها نتائج جيدة وممكن أنه نستخدموهم كتقنيات، معناها ما فيهمش مشاكل كبيرة. لكن طبعا الامبريوجينيسيس أسرع وأكثر، لكن يعني خرجنا بنتيجة أنه ربما البيرسونال هو اللي ممكن يأثر أكثر عامل يأثر على يعني عدم إنتاج جيد. وشكرا. وهذا معناها ذكرت أنت بصعوبة زراعة الأنسجة في نخيل التمر. نعرف أن مئات المختبرات فتحت في 2008 وبعد أغلقت كلها وبقوا يعدوا على الأصابع. فنريد رأيك أيضا في هذا الموضوع. شكرا.
Thank you, Dr. Abdullah. Dr. Mustafa, would you like to answer the question and then we will go to the next one?
Okay, thank you very much. Thank you الإخوان على الأسئلة. بالنسبة لدكتور عبد الباسط أسأل هل فيه.
If you allow me just one thing, we have English participant and they need.
Okay. Sorry. Yes. Dr. Abdul Basd asked about Sumatranal variation or any variation that can occur on TC plant, and he's asking if we observed some of this. So the question is, yes, you have Sumatranal variations can happen. In our case in Morocco, the most thing that we observed is an epigenetic variation that you can see when, but in a very, let's say, I don't have the statistic because nobody did the statistic, but very low appearance of some plant with the yellowish palms. You can see a palm and it looks like it's a nutrient deficiency, but it's not a nutrient deficiency. So it looks like more a physiological problem. And why I say it's epigenetic, because it goes with the time. So it starts usually, it might appear the second year, you might not see the first year, but the second year when the plant starts growing nicely, then you've got some of the leaves that are strangely yellowish, part of the leaf, in the middle of the leaf sometimes. And then in the third year, this will go and it will just disappear. So do you have this kind of things? Yes, that's what we observe. We occasionally also observe some plants which are with strange, let's say, phenotype, where the palms, for instance, do not look like they should be looking. They're a bit shorter, etc. The strange thing is, I followed one or two of these plants, and they did give fruits, and the fruit was prototype, but the phenotype was very strange. I mean, it was not looking like the right plant. So, but the occurrence of this phenomenon was very low. So I can follow up on the second question from Dr. Abdullah about how far can we go with DNA analysis, and how sure can we be that the plant is true to type using DNA analysis? Well, to start with, you know, I'm surprised that some people are still using RPD to analyze their plant, which doesn't mean anything. I mean, any lab who does not have a nice protocol, SSR is very efficient, but with condition. I mean, one of the first conditions, like I said, is that you have a database that it was done properly. How do you make your database? I mean, you cannot just go, let's say you are working on Majhul and just go anywhere and collect some leaves and say, I'm going to make my leaves, my database with this. No, that's not the way to go. You need to go to the field, adult trees, select 10 to 20 trees of the same variety. You know where that variety is so that you can trace it, go back to it, and then with that, run your markers, like let's say 20 markers, and with that you set you see that there is no differences to start with within that population, and then when you are sure that there is none and all the plants are similar for that given number of markers, then you can say, okay, I've got Majhul in my database, and I'm sure that it's a real Majhul, and that's what Majhul should look like, because you might have variation within the variety. Now, that's one of the first condition. The second condition, like I said, you cannot expect someone to say for sure that your plant is true to type using 1 or 2 markers. I mean, it's ridiculous. if you are sure, if you did the right running, that's why actually my plant, the adult plant, mother trees, I ran them with at least 19 different markers, because that's the base for me. I need to, that's my starting point. If I start with something wrong with or something with the Sumatranal Variation to start with, that's what I will propagate. So you need to be strong in terms of the number of markers you are using. You need to make sure that actually the markers you are using are really distinguishing between varieties, and you need to increase your numbers of markers to make sure that it's true to type. If you got all the conditions, yes, it's very robust. SSR, I believe in it, and I tried it. It's working perfectly, it's working nicely, but these are the conditions. And then Dr. Abdullah, he was asking about the human error. Of course, that's the problem. That's why you need the traceability system. You need a traceability system for two reasons, because people can make mistakes when subculture and cultures, and this happens. It's not nothing to do with technology. Whatever the technology you're using, people just can take two different jars of two different varieties and mix them, and you've got mixture of varieties. It's nothing to do with the technology, it's a human error. So when you are using the software, like what we do is when you take a batch and you screen it using your reader, if it's the wrong variety, it will go red and it will know this bottle or this jar is not the right variety, so you don't mix it. That's one thing. Another thing is, whatever the technology, whether it's organogenesis or embryogenesis, if you don't use it properly, if you propagate more than you need the buds that you have, whether it's organogenesis or somatic composition, you will end up by having some troubles for sure. So how do you do that? Again, traceability. So you know how long you keep a given batch in your lab, and after a period of time, you need to get rid of this plant. So the way to work in a commercial lab is to have waves of introduction. You've got continuous introduction of offshoots in your lab, so that all the offshoot will go out while the new offshoot is coming in, and you've got a series of production, and you've got waves. And so sometimes you see people introducing just once in 2 years or something, and say, okay, I don't need more because I've got plenty in the lab, which is wrong, because then you are in trouble, because you might over propagate or multiply your tissues, whether they are callus embryos or buds. I hope I answered the questions.
Thank you, Dr. Mustafa. Please, Dr. Rida Mizyani, you can ask your question.
السلام عليكم.
السلام عليكم.
شكرا دكتور عاد. الشكر الموصول للدكتور عبد الوهاب زايد وجائزة خليفة على هذا العرض.
وكذلك بس مش دكتور. تفضل.
ولا مشكلة. أيضا الشكر للدكتور أيد شيت مصطفى على هذه المحاضرة الأكثر من رائعة. أنا عندي 2 بوينت. النقطة الأولى بخصوص الكل يتحدث على الأورغانوجينيزيس والسوماتيك امبريوجينيزيس، وبأن السوماتيك امبريوجينيزيس ممكن أنها تعمل لنا السوماترانال فاريشن مقارنة مع الأورغانوجينيزيس، ولكن أنا كنظن النقطة اللي ممكن أن الدكتور مصطفى يشرحها أكثر ولا يبان أكثر، هو أنه حتى الأورغانوجينيزيس إذا ما كانش الاتباع ديال بعض المعايير اللازمة يكون عندنا فيها مشكل كبير ديال السوماتيك امبريوجينيزيس، لأن الفكرة ماشي هي اسم التقنية، ولكن كمية الضغط الممارس على الإكس بلانت ولا عليها. كل ما كان الضغط كثير خاصة عن طريق الهرمونات، كل ما عاد يكون عندنا مشكل ديال الأورغانوجينيزيس، لأن الكل يركز على التسمية وما يركزش على الطريقة المتبعة بالضبط في المختبر. ثاني نقطة للدكتور مصطفى إذا كان ممكن، لما نتحدث على ليزاناليز موليكولار على التحاليل الجينية، بالنسبة للإنف هذا شيء جيد، يعني نعرفوا واش تايب ولا لا، ولكن بالنسبة للسوماتيك فاريشن واش هذا الاناليز ممكن تعطينا فكرة ولا لا؟ هذا السؤال. شكرا جزيلا مرة ثانية.
Thank you for the question, Dr. Mustafa.
Okay, thank you, Dr. Rida. It's very interesting question. Actually, I remember where in the conferences where you have that confrontation of technologies, and people using organogenesis will say, oh, this is the best technology and the somatic embryogenesis is wrong. and people using organogenesis, somatic embryogenesis, they say, no, somatic embryogenesis is very good, and maybe organogenesis is not good. You know, the funny thing is, usually you find people doing organogenesis, blaming somatic embryogenesis, but they never used it. And the other way around, you find people using somatic embryogenesis, blaming organogenesis, but they never did. Well, maybe I'm lucky enough that I have used both. So to me, saying which one is best, it's like saying you've got, let's say, a brand of a car that it's more likely to make an accident than another. Yeah, it's true that you've got some cars are more robust than others, but at the end of the day, it's the driver who makes the accident. It's not the car. So it's the same with the technology. How do you use the technology? The somatic embryogenesis, yes, had some problems in the beginning, but these people using somatic embryogenesis, they're not stupid. They're very clever people. They adapted. They saw where was the problem, and they changed the protocol. So really, the technology is nothing wrong. The technology is what's wrong is the way people are using the technology. That's why I said tissue culture of date palm is very profitable and is very good if you do master the technology. And you master it, you need to master every single step, and everything has to be recorded, and you need to have a very well-trained staff that know exactly what they are doing. Otherwise, whether it's organogenesis or somatic embryogenesis, yes, like Dr. Rida said, you have trouble. Even in organogenesis, I mean, you cannot keep buds for ages, just subculturing them. Actually, you will notice that if you over subculture your buds, your rate of multiplication will go down. The quality of your plant that come out of those tissues is very bad. You have leaves that are not open. You've got bad roots, etc., etc. So you need to master the technology, and you need to have rules. How many times you will subculture this batch, and how we will handle that batch from starting point when you receive the offshoot until you deliver it to the farmer. So whatever the technology, if the science is not behind it, it's risky.
Thank you, Dr. Mustafa. Dr. Abdul Wahab Zaid may.
Yes, thank you very much.
The floor is yours. Thank you.
I just would like to mention here that I forget our dear colleague from Mexico who's joining us. So we have 15 countries so far. Thank you, Dr. Mustafa Rashid, dear colleagues. This is just the first conference on the subject. So we will have 5 more conferences that I will promise you it will be as energetic as this one. So we can say a lot of things, but at the end, I am very happy to see how things are going now with the tissue culture in general, regardless of technology used or technique used. But just for record purpose, Dr. Abdel Basit mentioned that Morocco is the only country using organogenesis. Yes and no. Yes, because it started there in collaboration with the French people. However, some scientists and technicians did move to the Middle East. So the UAE University laboratory in Al Ain is using organogenesis. And there is another lab in Saudi Arabia using organogenesis. So this is for record purpose, but we are not saying this one is better than the other one. We are just stating the information who's using what. So once again, I would like to thank Dr. Mustafa Rashid for such a nice, fruitful presentation. I would like to welcome, please feel free to send me directly or to Mr. Ahmed, any participant who would like, any scientist who would like to present something within this framework. We do have now 2 already selected conferences, so it will make it 3. However, we leave the door open to scientists to present something if they feel comfortable with the subject and they have enough material to present to their colleagues. Once again, thank you very much. It's 9 o'clock here in Amman. We have to go, so good night, everybody. Thank you.
Sorry, Dr. Abdul Wahab, just a small detail. I would like to remind الأخ عماد that he doesn't have the latest version. I made few corrections, so that will send him the latest version, the corrected version of the presentation.
He will receive the information, and we will task him to wait until we send him so that we can distribute it. But usually the second day we distribute, so please do your best to send it to him.
Definitely.
Thank you again very much. Good night, everybody. Wish you a nice evening.
Thank you for attending. Sorry for the participant which we didn't take their question. Looking forward to meet you in the next session about date palm tissue culture. Thank you.
إن شاء الله. السلام عليكم.
شكرا للجميع. شكرا للجميع.